Hello, I don't know if you can see me. Wait a minute. Oh, now I think you can see me. Hi, I'm going to record some videos for you to watch to learn about things like goblet cells, connective tissues, the integumentary system. I have some old videos about these things, but they are old and I can't find them. So let's get into Monday, October 6th, 2025. And let's see what we need to cover. Good luck. So our first topic was goblet cells, and I drew a picture of a goblet cell. So stop the video and remind yourself, what does a goblet cell accomplish? What is its function? And you should have come up with, it makes mucus and secretes it to the apical side of a epithelial tissue. And we find these all over the body. And under histology, what they look like, the reason I'm showing you this is what they have a distinct appearance under histology, where normally you see a row of epithelial cells that are colored in some way. They're pink or purple, and they have purple nuclei or some kind of dark nuclei. And then you'll see basically a colorless spot where it's the same as the background color. And that's because the mucus does not have any pigment. So goblet cells have that distinct upside down goblet appearance. Okay, so let's get into it. These are the four main tissue types. When we get into connective tissue, we have to subdivide it a lot. So we had epithelial tissues, and those are mostly named after their arrangement of cells because they are highly cellular. Connective tissues, which we saw, are tissues made of cells separated by extracellular matrix. And that matrix was made of fibers and ground substance, and that ground substance was made of interstitial fluid and proteoglycans. And below it, it's turtles all the way down. That's a science joke. Sorry. Another thing I brought up at the start of lecture were the two types of tissues that I skipped on Wednesday, and one of them is the pseudostratified columnar epithelium. And so the name kind of, what it gives you is pseudostratified. So it's not a stratified epithelium. It is one layer of cells, but the cells are very long in the basal apical direction. And what you see is that their nuclei are kind of displaced by other nuclei around them. The cells are very tightly squeezed together. Let's see, slides wise, there isn't necessarily cilia, because in the ducts of glands, they wouldn't need that mucus lining, but more common is the pseudostratified ciliated columnar epithelium, or columnar ciliated epithelium. I think this is how it's usually written. And you might be able to see under histology the thin little hairs. Those are the little cilia. And these are really nice pictures that have that layer of hair, this layer of hair. And what you're seeing is different stains applied to different tissues. But what these all have in common is they are the ciliated version of pseudostratified columnar epithelium. And whenever you see the cilia, there has to be a reason for it to be there. And in this case, it's usually going to be to push mucus as part of the mucociliary escalator. Let me say that again, mucociliary escalator. So and that's not something you need to learn, but that is an awesome thing to know. So I brought that up to say that if you're seeing the cilia, you should be also be seeing the goblet cells. And this picture has amazing goblet cells. The other tissue I showed today was transitional epithelium. And this is a cross-section of, a ureter. Right, there it is, a ureter. In the transitional epithelium, it's just that one tissue there, has this really folded look because it is collapsed on itself, and this could stretch. And if it stretched all the way out, it would be circular. So this epithelium, this layer, has this, it has this really strange appearance where at the apical side, the nuclei don't at all match the orientation of the cells that are more basal to them. So you have tissue that looks like a stratified epithelium and then some weird cells on top. And again, if you stretch this out, those weird cells would blend in a little better. So that is the transitional and pseudostratified columnar epithelia. So that brings us to the four main tissue types. And we looked at epithelial. Briefly, we'll look at muscle, nervous, and then we'll get into connective. So there are three types of muscle tissue, and they are very distinctive. They're nothing like epithelial. They're nothing like connective. They generate contraction. And these diagrams show the three types and the major differences between them. So things you can see in skeletal, cardiac, and smooth muscle. Well, what you might see in histology of skeletal and cardiac muscle are striations. And the striations are these vertical lines. And those come from the internal structure of the protein motors that generate the contraction. You can actually see them under histology. You can't see the individual proteins, but you can see the arrangement of large numbers of the proteins. And something notable about skeletal muscle is that the individual cells can be very long, and each one would have a lot of nuclei. Because when they're forming, they form from lots of small cells. So you could have a skeletal muscle cell the length of an entire muscle. And so you'd have lots of parallel muscle cells. Cardiac muscle, the cells are shorter, much shorter, and they branch to form a web that surrounds the heart. Cardiac muscle has what's called intercalated discs, and these are the connections between neighboring cells. So those dark discs. And sometimes those show up under histology and sometimes they don't. Smooth muscle usually to me looks kind of like a jumble, but what it aims to show is these spindle-shaped cells. And smooth muscle is found again in blood vessels, in skin, in the digestive system, really anywhere contraction might be necessary that is not under our direct voluntary control. So there are some pictures in the slides. This is an amazing picture of skeletal muscle showing striations and the parallel cells with the striations. I have never seen a picture this good anywhere else. Cardiac muscle, again, amazing picture showing the branchingness, the striations, the intercalated discs. And then smooth muscle. What I've never seen in smooth muscle is the spindle shape. This is a pretty common way for them to look. You'd have lots of kind of flattened nuclei that are parallel to each other. And each muscle cell has its one nucleus in smooth muscle. So this is histology of the three muscle types, and they this might be more what you'd expect to see. If you see the striations, you ask yourself, skeletal or cardiac? And if you don't see what looks like branching, and if you do see the nuclei are kind of on the edges, flattened on the edges of long fibers, then that's skeletal. If you see evidence of branching, so we do see some evidence of things like that. So a Y shape there, Y shape there, Y shape there. And cells like more nuclei like that, that would be cardiac. Smooth muscle is just hard. Because what you see are these long bands of contractile tissue that might or might not stain in an interesting way, and scattered nuclei. So that brings us to nervous tissue. And nervous tissue has two types of cells, neurons and neuroglial cells. And the neurons are gigantic. They have a huge, what's called cell body with projections or processes coming off of them. And we will look at that in this class. And the neuroglia are normal-sized cells with normal nuclei. So this is a zoomed out view, 100 times magnified, where this is probably more like 400 times magnified, just to show huge neurons. Nervous tissue would have these, you'd probably see neurons with processes coming off of those, and lots of smaller evidence of neuroglia around them. Oh, the other thing we skipped was membranes. We have cutaneous membranes, that's skin, and we have serous membranes. That's the membranes that separate our internal organs, like the membranes that separate our heart from our lungs, so the pericardium from the pleura. They're actually surrounded by double-layered membranes, and they have an interesting and complicated shape that we'll get into later. All these organs can't be completely surrounded by a membrane, because if they were completely surrounded, they would be cut off. And they can't be cut off from blood supply. They can't be cut off from the other ones they work with. So what we see is complicated folding shapes, where as the organ grows, it has a complicated double-layered membrane that it kind of grows into. You didn't see that coming because I didn't tell you about it, but that was on the list of things we skipped from Wednesday. Enough about that. So that was the end of Wednesday. In the next video, we will do connective tissues, and after that, the integument.